All right, so we're going to be starting chapter 9. Chapter 9 is a pretty long chapter. It's quite detailed, so it's going to be broken down into 3 parts. Be sure to look out for part B and part C. If you subscribe to the channel and if you're set up to receive alerts, you should be getting alerts as soon as the lectures are posted. Also, if you look in the comments below and also in the playlist for anatomy and physiology, you'll see all the chapters there too. Okay, so when you look at your muscles, muscle tissue, it makes up roughly half of your body's mass. Now, muscle, the other fascinating thing to know about muscle is that they do an enormous amount of work throughout our entire body. Some muscles, for example, they're relatively large, and some some are very small. So, for example, the smallest muscle you have, it's found in your middle ear. At the other end of the spectrum, if you want to think about the size to strength ratio, your tongue is the strongest muscle in your body compared to its size. When you're walking, you're using over 200 muscles, just to walk. Those are all the muscles that are used when you're in the process of walking. In addition to that, when you're frowning, over 40 muscles are used. When you're smiling, more than 11 muscles, about just around 11 muscles are used. So again, you don't think about it, but muscles are doing a lot of muscles are working together to bring about motion, whether it's to do work or to make expressions of how you're feeling. Muscles, most of the muscles, they work together in groups. And you will see that as you progress through. More so, you'll see it in chapter 10. So now, what exactly do muscles do? Well, muscles, they work by contracting. When they contract, they exert force. And how they're able to do this, how muscles are able to exert force, is that they have the ability to transform chemical energy into a directed mechanical energy. Now, that chemical energy is ATP. Remember, adenosine triphosphate. Now, to look at muscles, we're going to be looking at the different type of muscles that we have. So we'll be talking about the 3 different type of muscle tissues we have. We're also going to be looking at the characteristics of muscle tissue. In addition to that, we're going to be looking at the functions of muscles. Now, when we're discussing muscle types, we're going to be seeing terminology such as myo, myos, sarco. These are all word roots that are referring to muscle. So, for example, if you see the term like myocardium, they're talking about heart muscle. Cardi is heart, so myocard. That's what we're talking about, heart muscle. You'll see the term mys as well, or sarco. So, for example, you might see sarcolemma or sarcoplasm. Sarcolemma, that's going to be the cell wall for a muscle cell, and sarcoplasm is going to be the cytoplasm of a muscle cell. We're going to be looking at the 3 different types of muscle tissues. So now these muscle tissues, there are skeletal muscle, which you find attached to bones. Cardiac muscle, the only place we find that is in the heart. And then we have smooth muscle. Smooth muscle, you find them surrounding hollow organs. Now, only skeletal and smooth muscles, they're elongated, which is why we call them muscle fibers. Now, skeletal muscle tissues are packaged into skeletal muscles, and they attach to your bones, and also you're going to find them attaching to skin. So if you think back to chapter 5 on skin, and we found those erector pili muscles there, and so when you get the goosebumps, those are the muscles that are responsible for it, and now you know that it's a skeletal muscle. The skeletal muscle fibers, these are the longest of all the muscles we have. And when you look at them in appearance, they have these stripes on them. And we refer to these stripes as striations. In addition to that, we're able to consciously control your skeletal muscles. So because of that, we call them voluntary muscles. Now, even though these are voluntarily controlled, there is some involuntary actions that still affect skeletal muscles. And that's primarily through the reflex arcs that we have in our body, which we'll be learning about later on as we go through the chapter. Your skeletal muscles are responsible for your overall body mobility. It's able to contract rapidly, however, it does tire itself quite easily, so it has to take some rest after a short period of activity. However, it still is able to exert a tremendous amount of power. Keywords for skeletal muscles include skeletal, striated, and voluntary. Now, cardiac muscle, the only place we find it is in our heart, and it makes up the bulk of the heart's walls. It is striated, as we saw in the case of skeletal muscle, but unlike that, it's involuntary. We do not have conscious control of it. your heart, it has its own pacemaker that we called the SA node or the sinoatrial node and the AV node or the atrioventricular node. And both these guys, they help in maintaining a rhythm in your heart. And your heart, it'll beat anywhere from, in a normal person, anywhere from 60 to 90 times a minute. When you go above that or below that, then it may not necessarily be a good or a bad thing. Now, if you go, for example, when you look at certain athletes, their resting heart rate could be as low as 50 beats a minute. Okay, it might be in some cases even lower. So for them, it's okay because, they may be runners, for example. So every day they're out and running, God knows, 7, 8, 10 miles a day. And again, you'll tend to see this in people that are athletic, people that run a lot or they do a lot of aerobic exercise. Their resting heart rate is much lower, and that's a good thing. Now, keywords that we're going to be seeing for this muscle type is cardiac, striated, and involuntary. Now, other thing I want to let you know about heart tissue. Now, notice that I said that the heart, it's not consciously controlled. Now, however, the nervous system, it can affect it when need be. So if you get very excited or if you have to run somewhere really far, your heart's going to make it work much faster. Again, if you get scared, again, nervous system sending signals to the heart, okay, start getting pumping, beat faster, because you might be, you may have to start to run. So it starts to get, things going a little bit. Again, just to increase your likelihood of surviving some type of a traumatic event that may be unfolding. A smooth muscle tissue we find lining the walls of hollow organs. So, for example, you'll find it around your stomach, around your bladder, your urinary bladder, and your trachea, okay, your airway. Smooth muscle is not striated, okay? So that's why we call it smooth, as the name implies. Now, smooth muscle also, it's involuntary, meaning we don't have conscious control of this type of muscle. It's able to contract on its own without your nervous system. However, the nervous system, it does have an effect on it. So in this illustration here, table 9.3, you're able to see the different type of muscle tissue and where we find them in the body and what they look like. So for skeletal muscle, you find them attached to bones, and also you'll find them attached to skin as well. As I said, we spoke about earlier in chapter 5. We also have cardiac muscle that we only find in the heart, and then we have smooth muscle where we only find lining the walls of hollow organs. Now, when you look at the pictures of this, when you look at the slides, the histological of these three tissue, notice that both your skeleton and cardiac, and you can see the illustration as well, is that they are striated. They have these lines, all right? And whereas smooth, it does not, which is why we call it smooth. The cardiac muscles, they're binucleate. It could be uninucleate, but your skeletal muscle, it will be multinucleated. And your smooth muscle, on the other hand, it will be uninucleated, meaning it will just have a single nucleus. Whereas your heart muscle, it'll usually have one or two nucleus. And then your heart muscle, it has multiple. It'll have many, 4, 5, 7, 8. However, it'll be more than 3, more than 2. Now, although we have 3 different type of muscle tissues, they all share the 4 main characteristics. And the first one of those is excitability. Other terms for excitability that we use could be responsiveness or irritability. Now, this is the ability for muscles to receive and to respond to stimulus. And the stimulus, it could be any change in the environment, either on the inside or the outside of the body. When we're talking about contractility, we're referring to the muscle's ability to shorten forcibly when adequately stimulated. Now, this ability is what sets muscles apart from all the other types of tissues. When we're talking about extensibility, we're talking about the muscle's ability to be stretched or extended. Muscle cells, they shorten up when contracting, but they can be stretched even more than their resting length when they're relaxed. And when we're talking about elasticity, now this is the ability for the muscle cells to recoil and resume its resting length after it's been stretched. Four important functions that muscles serve. The very first one is to produce movement, and this is responsible for not only for local motion but also for manipulation within the body. So, for example, when we're walking, okay, we're moving about. You're lifting your arms up and down, or you're swimming. This is all helping you move about within your and interact within your surroundings. But in addition to that, you're from within the body. When we talked about smooth muscle, your smooth muscles lining the walls of your stomach, so it's helping in digestion. You find them within your intestines as well. You also find them in surrounding your blood vessels, and they contract and they relax to help control your blood pressure and also to increase or decrease pressure in certain areas, or in returning back to the heart. In addition to that, we have your heart, which is the main, your cardiac muscle, which is the main pump that is circulating the blood. So again, it's very extremely important function for muscles to be able to contract, to bring about all this, both locomotion and manipulation. Now, while these are vital, when you look at maintaining posture in your body position, you don't think about it, but your muscles, skeletal muscles, they're doing this even right now without you being consciously aware of it. You have certain muscles that are contracted, others that are relaxed. That'll help maintain your body position and your posture. The same thing when these muscles are contracting, they're also helping to stabilize joints, as you learned in the previous chapter, in chapter 8. We also talked about very earlier on that when you're shivering, one of the reasons that you're shivering is because your muscles, when they're contracting, they produce heat. So when muscles contract, it does produce heat, and this helps maintain this body temperature that we like within this range. So we like to stay warm. Our body temperature needs to be right around 98 degrees, 98.6. So we're right around 98.3, 98.4, 98.5, 98.6, 98.7, 98.8. We're good. When we start going above 99, then you might start to feel slightly sluggish. But then again, when you're exercising, again, your body temperature does rise. And again, we don't feel very comfortable at that point either. So which is why we need to cool down. In addition to that, other things, aside from these very four important functions, muscles, they also help protect your organs. So think about your liver, for example. While there is part of your rib cage that's help protecting it, your muscles, you have a lot of muscles there as well that are help protecting that liver. In addition to that, if you think about, for example, your bowels, your large intestines, there is no rib cage that's over there. There's no bones that are protecting it. The only thing that's there to protect it are these muscles. They also form valves, for example, within your heart. You have these valves that are there that open and close to let the blood go from one chamber to the other and to the other. Around your eyes, they help control the size of your pupil, which causes the amount of light that enters is able to enter the retina. Again, we talked about goosebumps. We talked about these erector pili muscles that are responsible for that as well. So when we look at skeletal muscles, it's made up of different tissues and has three unique features. First of all, skeletal muscle has a very rich nerve and blood supply. In addition to that, we also find connective tissue that your skeletal muscles is wrapped within. And also, skeletal muscles, they attach to bones. So you'll find them having an insertion and an origin points of bones. And think back to the previous chapter, in chapter 8, when we talked about the insertion. That's going to be the bone that's going to be moving, and then the origin, that's the bone that's not going to be moving, or it's going to be the bone that's more stable. Generally speaking, each muscle is served by one nerve and artery and by one or more veins. These structures, they all enter or they exit near the central part of the muscle, and they branch profusely through its connective tissue sheath. Unlike your heart and smooth muscle tissue, which are able to contract in the absence of nerve stimulus, each skeletal muscle fiber is supplied with a nerve ending that controls its activity. Now, skeletal muscle, it has a rich blood supply, as we said earlier. This is understandable because contracting muscle fibers, they use a huge amount of energy and require more or less continuous delivery of oxygen and nutrients via the arteries. Now, muscle cells, they also give off large amounts of metabolic waste that has to be removed through the veins if contraction is to remain efficient. Now, each skeletal muscle, as well as each muscle fiber, is covered by connective tissue, and it's there to help support the cells as well as reinforcing the muscle as a whole. So think of it this way: it's preventing the muscles from like bulging out and separating when the muscles are contracting. Now, we're going to be looking at these different types of connective tissue going from external to internal, or these different sheets that we have that make up this connective tissue sheet. So the outermost layer is this epimysium. Now, this is made up of dense, irregular connective tissue, and again, it surrounds the entire muscle. Now, sometimes it blends with the deep fascia that lies between the neighboring muscles or the superficial fascia that's deep to the skin. Next, what we have is the perimysium and the fascicles. So within each skeletal muscle, the muscle fibers that are grouped into fascicles, or these are like, think of them as bundles. So if you look at it in the diagrams and the illustrations that we'll see in a handful of slides, they may resemble like a group of sticks that that are held together. Now, surrounding each fascicle, we have a layer of fibrous connective tissue that's called the perimysium. So remember, first we had dense irregular connective tissue. Now we have fibrous connective tissue. Now finally, when you go to the next type of connective tissue that we're going to find in this next sheet, in the endomysium, we find areolar connective tissue here. So in the endomysium, this is a sheath of connective tissue that surrounds each muscle fiber. So when we look at this illustration here, what we're seeing is, first of all, it's a cross-section of a muscle, and this entire muscle over here, okay, it's surrounded by this epimysium, right? And then this is the dense irregular connective tissue that we have. Now, each one of these things that you see over here, okay, this, and this over here, which has been pulled out, this is what we call these fascicles. So this entire fascicle, it's wrapped by a perimysium. Okay, so this perimysium is wrapping this entire fascicle. Now, when we look at these individual muscle fibers, okay, that makes up this these fascicles, what we see is that it's lined by a endomysium. So this lining here, this is an endomysium, and you can see this lining over here. See, over here, over there, over there. This is all the endomysium that's running around these muscle fibers. So from innermost to outermost, what do we have? Endomysium, perimysium, and then we finally have the epimysium. So when we look at muscles, keep in mind that muscles, they go across joints and they attach to bones. Now muscles, they attach to bones at 2 points. We have the origin and insertion. Now this is back from the previous chapter, in chapter 8, and remember what we said at that point. We said that the bone that's doing that's not going to be moving or moving less, that's the origin, and the bone that's going to be moved closer to the origin, that's going to be referred to as the insertion point. Okay, so once again, origin and insertion. The bone that's going to be moving, that's the insertion, and the bone that's not going to move or does very little moving, that's the origin. So when you're looking at, for example, your forearm and your upper arm, okay? So your radius ulna and your humerus. And when you flex at the elbow joint, and again, we're looking at your biceps muscle. So which muscle is, which bone is moving? Well, the bones that, or which bones are moving, I should say, are your radius and your ulna. So these bones are moving. So therefore, the insertion point of the biceps is going to be on the radius and ulna, right? And then the origin, which bone is not moving, that's going to be on your humerus. Okay. Now, going down over here, when we're talking about attachments, they could be either direct or indirect. So when we're talking about direct attachments or fleshy attachments, what we're referring to is when the epimysium it's fused to the periosteum of the bone or the perichondrium of the cartilage. We also have these indirect attachments as well. Now, in this case, the connective tissue wrapping, it extends beyond the muscle, and it acts like as these extensions that we call tendons, or it could be this sheet-like aponeurosis. So let's go back over here again. We already looked at this diagram, this illustration. So let's go over here. Now, this is a pretty good job of summarizing what we spoke about. So this is your entire muscle. What did we say? The entire muscle is covered by this epimysium, and we said that this entire muscle, it's made up of these fascicles. All right. So, and as in the description it says, the muscle, it's made up of hundreds of thousands of muscle cells plus connective tissue wrappings, blood vessels, and nerve fibrings. And again, what do we have covering the entire muscle? We have this epimysium. Now when you go down over here, now remember what we said. This entire muscle is made up of these bundles of there, fascicles. Okay, and this is what this is. This is a fascicle. So now let's take a close up of this fascicle. And this is just a cross section of a part of a fascicle. And what we see is that the fascicle, it's lined by this membrane, which is called this perimysium. All right. So now, so this fascicle, it's made up of a bundle of cells. Again, that's separated by this perimysium. So over here, this perimysium separates this other bundle of cells. The perimysium here, it separates this fascicle from this fascicle here by, again, by the connective tissue. And then we go down to this photo over here. So now what we do is this. We looked at this the fascicle. Now let's pull this out and see what these muscle fibers are made up of. So, which is where we come down over here. So this entire fascicle is made up of these muscle fibers, right? And this is what we look at, this muscle fiber cell. And what are these muscle fiber cells made up of? They're made up of these myofibrils, all right? So the muscle fiber cells, now they're lined with endomysium, okay? So there, over here you can see the endomysium over here, all right? And then you can see the myofibrils over here. Now we're going to be looking at these things in more of a detail at this point, at this level, as we move forward. Now, skeletal muscle fibers are long cylindrical cells that contain multiple nuclei. Now remember, we're going to find this in skeletal. In smooth muscles, it's going to be different. It's going to be a single nuclei, and it's not going to be striated. And same thing for cardiac muscle. It's going to be striated, but it's going to be binucleate. Also, we have a sarcolemma and sarcoplasm, and we mentioned these a little bit earlier. So the sarcolemma, this is the plasma membrane for the muscle fiber, and the sarcoplasm, this is going to be the cytoplasm for the muscle fiber or the muscle cell. Now, these terms, muscle fiber, muscle cell, we use interchangeably. We also find them containing glycosomes. Now, glycosomes are there for glycogen storage. And remember, what did we say that muscles, they need a lot of energy? Remember, what do? They take ATP and they convert it into contractile force. So their source of energy, or the fuel that it needs, is ATP, but then it can also come from we'll talk about later, but these glycogen, for example. So again, it needs sugar. So that's why these glycosomes are also contained there. In addition to that, we find a lot of myoglobin that's going to be present there as well. And the myoglobin is there to store oxygen. And again, if you guys think back to chapter 3, you need oxygen for the Krebs cycle to take place. And in order for ATP to be produced, you need to have oxygen present. We're also going to look at organelles that are found over here. So these include the myofibrils, the sarcoplasmic reticulum, as well as these transverse tubules, or sometimes they're referred to as T-tubules. Now, myofibrils, these are densely packed rod-like elements, and a single muscle fiber, it could have thousands of them, and they account for roughly 80% of the muscle cell's volume. Now, we're going to be looking at these features of this myofibrils, and these are important. So we're going to be talking about the striations, what they mean, the sarcomeres, the myofilaments, and also the molecular composition of myofilaments. So when you're looking at this illustration over here, we have these dark bands, which we call these A bands, and then you have these light bands, which you call the I bands. And this is the nucleus. Once again, this is a myofibril, and you can see dense. So look at how densely they're packed. Now, in between them, you see these mitochondria. And remember, what is the mitochondria? It produces the energy for the cell. Now the striations, they're formed by a repeating series of dark and light bands, and you can see this along the length of each myofibril. Now in intact muscle fibers, the dark A bands and the light I bands, they're nearly perfectly aligned with one another, giving the cell this entire striated appearance. Now, each dark A band, it has a lighter region in its midsection. That's called the H zone. And each H zone, it's bisected vertically by a dark line called the M line. That's formed by molecules of the protein myomycin. Now, the light I band, it also has a midline interruption. And this is the area, it's a darker area, that's what we call the Z lines. So when you look at this slide over here, what we see is we see these dark bands over here, and so these are the A bands. You can see the nucleus over there. There is another one here and there, and then you see these lighter I bands. Okay, so you have I bands, and then you have these darker A bands, and then you have these muscle fibers here, muscle fibers there. So a sarcomere is the smallest contractile unit of muscle fiber. So they're functional units of the muscle cell. The sarcomere, this is a region of the myofibril that we find between the two successive Z discs. So it contains an A band that's flanked by half an I band at each end. Now within each myofibril, the sarcomeres, they're aligned end to end, kind of like in a boxcars of a train. Now this is a very important picture or illustration to look at and to understand. To go over what we just talked about, because we're going to be referring to these over and over again. So you really need to understand this part. This is very important. So what you see over here are these dark lines and then these light lines. And these dark lines are these thick filament that we talked about, okay, or the myosin filaments. And these blue colored, these light bands, these are called the thin filaments or the actin filaments. Right, so you can use this interchangeably. Thick filament, you can call it myosin filament. Thin filament, you can call it actin filament. So now, again, so remember, this is a sarcomere, all right? So what you look at over here, when you see the area of the stretch of an entire thick filament from one end to thick filament another end, this is called the A band, okay? So A band goes from one end of a thick filament to the other end of the thick filament. That's the A band, all right? Now, from the part where the thick filament is absent from one end to the other end, that's referred to as the I band, okay? I band. So remember, what do you have in I band? I band, it lacks thick filament. I band only consists of your actin filament, only myofilament, thin filament. That's what the I band consists of. Now, let's look at what the Z disc is, okay? So the Z disc is where these I bands, they lock together, okay? They link up together. This is what the Z disc is. So where you have these thin filaments, I should say, where the thin filaments, they end up linking up, thin filament from one end, thin filament come from another end, they end up coming together, linking up, then it forms the Z disc. So we now spoken about the Z disc, okay? And this is the Z disc here, and the part where you have only thin filament, all right, and no thick filament is called the I band. And then we said the A band is a stretch that goes from one end of the thick filament to the other end of the thick filament. Now, in between, in the middle of the thick filament, so in the middle of the A band, where there is not any thin filament present, we have this H zone. Okay, so the H zone, first of all, it lacks thin filament, and secondly, you see these protein chains running vertically at a 90-degree angle. And remember what we said this was earlier, is we call this protein myomycin. And remember, this is the part, this is what forms this M line, the myomycin. And the area where you find this myomycin and it lacks that the thin filament is this H zone. Okay, so what do you have within the H zone? Within the H zone, you have myomycin. Why do we have myomycin? Because it lacks the thin filament. All right. So once again, make sure you guys go over this and make sure, on exams, expect questions to see like this, like where is the point where the thin filaments, they connect with one another. So, that's going to be the Z disc. Or again, what do you call the line where they end up meeting together? And what's the area where you have only thin filament present? When you have thin filament present, that's going to be your I band, or which band? And then another question could be, where do you find only thick filament and no thin filament? So, thick filament and no thin filament, you're going to find in the H zone over here. All right, so think about these type of questions when you're studying this and looking at this. Now, these myofilaments that we spoke about in the previous slides, they're orderly arrangements of actin and myosin filaments within the sarcomere. So the actin myofilaments, these are the thin filaments, and they extend across the I band and partway in A band. They're anchored to the Z discs. Now, the thick filaments, or the myosin myofilaments, these extend the length of the A band, and they're connected at the M line. Now, when we look at the cross-section of a sarcomere, we'll see a hexagonal arrangement of one thick filament that's surrounded by 6 thin filaments. So when we look over here, you can see this centrally located this myosin filament that's surrounded by this an arrangement of this hexagonal arrangement of 6 thin filaments, or the actin filaments. So when we look at this picture over here, notice we see this, and we haven't seen this before. Now, these are these titin filaments. These titin filaments, they run through the core of the thick filaments, and they're anchored onto the Z discs. So if you recall back when you look at this slide, these run over here, okay? So, just because they're not pictured over here doesn't mean that they're floating magically on their own. No, that's not the case. Those titin, those elastic filaments is what's keeping them in place. So yes, now, so this is your Z disc over here. And then so again, let's just compare it over here. So you have your Z disc over here to what we saw over there. And again, you cannot see these the titin filaments over here, but this is where they're anchored onto. Now let's look at the next area back over here. So now you have this the M line over here. So remember, in the M line, what you have is these protein that's going perpendicular to the thick filaments, the myosin filaments over here, and this makes up this H zone. So the H zone is where you have only the thick filament, and you don't have any thin filament. There's no thin filament over here in this H zone. And what else can we see? So yes, so this area from here to here, this is your A band, and then this will be your the area that's running from this end all the way to where the next your next myosin band your next A band starts. That's going to be the area for this I band that we'll find. We're going to be looking at the molecular composition of myofilaments now. Now, in the thick filaments, they're made up of the protein myosin. Now, myosin, it's made up of 2 heavy and 4 light polypeptide chains. Now, the heavy chains, they twist together to form the myosin tails. And the 4 light chains, they end up forming that globular head of myosin. Now, during contraction, the heads, they link thick and thin filaments together. And this is what forms this cross bridge, as we'll see as we move through the lecture. Now, the myosin molecules, they're offset from one another, and this results in a staggering array of heads at different parts along the thick filaments. Now, in thin filaments, it's made up of the fibrous protein actin. And actin is a polypeptide made up of these kidney-shaped G-actin subunits, or these globular subunits. So these G-actin subunits, they have these active sites that are the myosin heads are able to attach to during contraction. So these G subunits, they link together with one another to form this long fibrous F-actin, or this thin filament, in other words. So when you have two of these actin strands, 2 of these F-actin strands, or these filaments that end up twisting together, they end up forming this thin filament. Now, we also find tropomyosin and troponin. Now, these are regulatory proteins that are bound to actin. And the troponin now is what the calcium is going to be able to bind to. And the tropomyosin is the part that's blocking the active binding site on this actin strand. So the myosin head is not able to attached to it. And we'll see this in the slides in some illustrations as we move forward. So here you go, right over here. So this is if you notice, if you look at the structure, this kind of looks like a golf club. So like a double golf club. So this is a tail, and notice how they're twisted together. And then you have these heads. Now, on the heads, you have these actin binding sites, and then you have these ATP binding sites, which you'll see later on in the role that they play. In this thin filament illustration here, these are these subunits, okay, these actin subunits. So the G-actin units that we were talking about. And when you look at them, they link together. And when you have two of these strands that are linked together, it's two of these filamentous, this is what forms the thin filament. So, and again, this is a cross-section. This is a small portion of a thin filament. And you see what we find over here are this tropomyosin and troponin. Now, troponin is responsible for binding of calcium, and then you have this tropomyosin that binds the active sites for these myosin heads to attach to. So essentially, what ends up happening is when the calcium attaches to this troponin, it ends up changing the shape, and then in return, what ends up happening is these tropomyosin, it gets moved off these binding sites, and then the actin heads are able to come and attach to it. And in this picture over here, it shows the myosin heads form these cross bridges over here. So here, this is the myosin head, and this is the thin filaments over here. So you can see these cross bridges that are forming. Again, the heads of the thin filaments. And again, this is a head, it's a thin filament, and these are the tails for the myosin. Other proteins, they help form the structures of the myofibril. Elastic filament, for example, it's made up of the protein titin. And elastic filaments, it holds the thick filaments in place. So if you remember, we went back a handful of slides, and you saw the thick filaments, and it was just kind of, floating in the middle. And then I told you it's not, magically floating. And this is where those titin plates are. It ends up forming the core of the thick filaments, and then it goes and it anchors onto the Z lines. The titin, it helps recoil after stretching. It also helps resist excessive stretching. Another important structural protein is dystrophin, which links the thin filaments to the integral proteins of the sarcolemma. Other proteins that act to bind filaments or sarcomeres together and maintain their alignment, they include nebulin, myomyosin, and C proteins. Now, if you remember myomyosin a handful of slides ago, we find those in the H zone, and they end up forming those M lines. Now, the sarcoplasmic reticulum, these are a network of smooth endoplasmic reticulum tubules that surround each myofibril. Now, most of them, they run longitudinally along the myofibrils, communicating at the H zones. Others that are called terminal cisterns, they form larger perpendicular cross channels at the A band and I band junctions, and they always occur in pairs. Now, closely associated with the sarcoplasmic reticulum, we have large numbers of mitochondria and glycogen granules. Now, both of them, they're involved in producing energy that's used during contraction. And the major functions for the sarcoplasmic reticulum is its ability to regulate the intracellular calcium levels. In so doing, it stores and it releases calcium when the muscle fibers are stimulated to contract. Now, at each AI band junction, the sarcolemma of the muscle cells, it protrudes deep into the cell's interior. It ends up forming this elongated tube that we call the T-tubule, or transverse tubule. Now, this T-tubule, it tremendously increases the muscle fiber's surface area. The lumen of the T-tubule is continuous with the extracellular space. Along its length, each T-tubule, it runs between the paired terminal cisterns of the sarcoplasmic reticulum. Then what this does is it forms these triads. And these triads, they're groupings of three membranous structures, which are the terminal cisterna, the T-tubule, and the terminal cisterna. Now, as they pass from one myofibril to the next, the T-tubule, it also encircles each sarcomere. The roles of T-tubules and SR in providing signals for contraction are tightly linked. At the triads, where these organelles come into closest contact, something that resembles a double zipper of integral proteins protrudes into the intermembrane spaces. The protruding integral proteins of the T-tubules, they act as voltage sensors. Those of the SR, they're called foot proteins, and they form gated channels through which the calciums can be released from the SR cisterns. When an electrical impulse passes by, the T-tubule proteins, they change shape, which causes the SR proteins to change shape. And this is what causes the release of calcium into the cytoplasm. All right, so we've seen a lot of terms in a handful of slides previously. So let's look at all this stuff graphically to help simplify matters. So again, we're looking at a muscle cell over here. And we have this is the cell wall, or the sarcolemma of this cell. And as you see, in continuation with the sarcolemma, you have these over here, and then you have another one over here. And these are these transverse tubules that we have. So these T tubules, they're continuation of the sarcolemma that run deep into the cell. So, and these are the myofibrils that we have. And as you see over here, these T tubules, they're surrounded by, on both sides, these enlargements of this sarcoplasmic reticulum. So this is the sarcoplasmic reticulum. Again, in a normal cell, in a non-skeletal muscle cell, you'd call it an endoplasmic reticulum, but since we're looking at skeletal muscle, these are called sarcoplasmic reticulum. So these enlargements that surround this T-tubule, these are called the terminal cisterns. Okay, so you have a terminal cistern here and another terminal cistern there. Now, the combination of the terminal cistern, one on this side and one on this side, and then in addition to the transverse tubule, this is what forms this triad. Okay, so what is a triad? The triad is essentially just the transverse tubule, or the T-tubule, and the two terminal cisterns. And then as you see going across, you have these the tubules of the sarcoplasmic reticulum, and again, this is all sarcoplasmic reticulum over here. And then you can see at this AI band junction here, this is where you start to see the terminal cisterns being found over here. And then also you have the Z discs over here, you can see. And then you have the M line right over here, and this is your A band that spans this length from one end of the thick filament to the other end of the thick filament, or the myosin filament. And then when you look at where there's no myosin filament, only you have actin filament, you have this I band that forms. All right. Then you have the Z discs over here, and another Z disc over here. And what you don't see over here is the titin filaments. Now, the term contraction, it refers only to the activation of the myosin cross bridges, which are the force generating sites. Now, shortening occurs if and when the tension generated by the cross bridges on the thin filament exceeds the forces opposing shortening and pulls the thin filament towards the M line. Now, contraction ends when the cross bridges become inactive and the tension declines. In the relaxed state, thin and thick filaments, they overlap only slightly at the ends of the A bands. Now, the sliding filament model of contraction, it states that during contraction, the thin filaments, they slide past the thick filaments, and this is what causes the actin and myosins to overlap more. Now, neither the thick filament nor the thin filaments change length. They just overlap more. They're not getting shorter or lengthening. They just start to overlap more, and we'll see that in the pictures when we move forward. Now, when the nervous system, it stimulates muscle fibers, the myosin heads, they're allowed to bind to actin. And this is what's the term that we use, saying that a cross bridge has been formed. And this is what causes this sliding process to start. Now, these cross-bridge attachments, they form and they break several times. Each time they pull on the thin filament a little closer to the center of the sarcomere. So it's kind of like a ratcheting action. So as this occurs over and over again in sarcomeres throughout the cell, the muscle cells, they start to shorten. Now, what you're going to see is the Z disc, which is starting to get pulled towards the M line. The I bands, they start to shorten. The distance between the Z discs is reduced. The A zone disappears. And the A bands, they move closer together, but they don't change in length. So when you look over here, again, what they're trying to tell you is this. So this is a fully relaxed sarcomere of a skeletal muscle fiber. So remember, what are they saying? They're saying that the myosin and the actin, it doesn't change length when it's contracted or relaxed. So when you're looking over here when it's relaxed, notice that only the ends of the thick filament and the thin filament, they only overlap towards the ends of this A band. Okay, everywhere else it doesn't. Now, when you look over here, now also, so again, let's here. I'm gonna bring this in, and just so you guys see. Okay, so look, this is the length of this thick filament when it's relaxed. All right. Now, in the next picture, it's gonna show when the muscle is contracted. So let's look over here. This is the distance, all right? And then if you look at the thin filament, it spans off right to where this F is on F9, okay? Roughly right over there. So we're going to go over there and we're going to see in the next slide. So here we go. Next slide here we are, guys. And if you look at now, this is so again, this is a picture of fully contracted muscle fiber. So look, the thick filament, it has not changed in length, all right? And when you look at the thin filament, remember we said it is from here, it's right at where the F is at the F9. And again, so rough, it's exactly the same. There's no change in length. If you look over here, you look over here, here, right over there, and then this same goes for this one, right? So again, what they're saying is this: regardless, there's no change in length. Now, what happens is this: the Z discs, they're pulled closer together. See, they're much farther apart over here, but now when it's contracted, they're closer together. The H zone is gone. We don't have this H zone anymore. See, you have this H zone over here. That's no longer there because you have both. Now you have overlap of both the actin and the myosin occurring. And yeah, I think that's all that it showed. This is pretty much this is what they're trying to tell you. So what happens is this: these guys over here, these are the myosin heads, and what they do when they start to contract, or again, they're attaching themselves onto this myosin, and then they pull this myosin closer towards this M line right over here. Okay, so they attach on here, and then they start. Think of it this way: this guy is going to attach over here, and is going to yank this actin thin filament close more this way. This guy's going to be pulling it in this way. So as they're doing it, one it attaches, it pulls it in. Another one's holding it in place. Another one comes up, it attaches, and then it pulls it in. The other one lets go. So this is how it happens over and over again. This is what they're talking about, this ratcheting effect. And the end result, when they do this over and over again, they end up pulling this the thin filaments towards the this M line, and then this is how the Z discs end up coming closer to one another. So and then overall you end up getting a shortening of the muscle cell. Now, 4 steps need to occur for skeletal muscles to contract. The very first thing that you need is a nerve stimulation. The second thing that needs to happen after the nerve stimulation is you need to have an action potential or an electrical current that has to be generated in the sarcolemma. The third thing is this action potential, it needs to move along the sarcolemma. And the fourth thing is you need to have intracellular calcium levels to rise. Now, steps 1 and 2, they occur at the neuromuscular junction, and steps 3 and 4, they link the electrical signal to contraction. So we refer to this as this excitation-contraction coupling, or EC coupling. Now, this flowchart, it goes through this series of steps that occurs in order for contraction to take place. So in the first phase, they broke it down to 2 phases, phase 1 and phase 2. So in phase 1, an action potential arrives at the axon terminal at the neuromuscular junction. As this action potential arrives at this neuromuscular junction, it ends up releasing acetylcholine. When acetylcholine is released, it leaves this the axon, and then it goes and binds to the receptors on the sarcolemma. Now, at this point, it changes the ion permeability of the sarcolemma. Once that happens, the sarcolemma depolarizes. When it depolarizes, you end up getting an action potential that's generated. Now, this action potential, it needs to travel across the entire sarcolemma. When it's doing that simultaneously, the action potential is also traveling along the T tubules. Now, when it's moving along these transverse tubules, it ends up releasing the calcium that's stored in the sarcoplasmic reticulum. Now, the calcium, it goes, and then it binds onto the troponin, and then it exposes the myosin binding sites on the actin filaments. Now, once these the myosin sites have been exposed, the myosin heads are able to go and bind onto the actin, and then this is when you get contraction to start. Now let's look at the nerve stimulus and the events that take place at the neuromuscular junction. Now, skeletal muscles, they're stimulated by somatic motor neurons. Now, axons, these are the long thread-like extensions of the motor neurons. These axons, they travel from the CNS to the skeletal muscles. Now, each axon, it divides into multiple branches as it enters the muscle. Now, axon branches end on the muscle fibers, and they form this neuromuscular junction, or another term that we use is motor end plate. Now, each muscle fiber has one neuromuscular junction with one motor neuron. So if you look over here, this is an axon, and it's coming. This is a muscle fiber over here. So this is this axon terminal over here, and so we call this the neuromuscular junction. Okay, this area we're looking at here. This is the neuromuscular junction. So we're going to be looking at all these steps. So this first step essentially is saying this: an action potential or a nervous stimulus. It comes down through this axon, and it ends at the terminal, the axon terminal over here. So when this action potential it arrives, you see these over here. These are these voltage gated channels, and when the action potential arrives, it knocks these channels open. Once these channels open up, calcium enters. Okay, this axon. So once the calcium enters the this axon terminal, what's going to end up happening is this. These vesicles you see here, they're filled with acetylcholine. Acetylcholine is a neurotransmitter. Now, when the calcium enters, these acetylcholine, it gets released through exocytosis, and it flows to this over here, which is we call these the junctional folds of the sarcolemma. Now you see on the junctional folds of the sarcolemma, you have these ligand gated channels. You see, these are the things that are in this purple, I guess, color. And these, so the acetylcholine is going to bind to these ligand gated channels, and it opens it up. Once it opens it up, then sodium is going to enter. Then it causes depolarization to take place. Then you get an action potential that's generated, and it's going to move across this sarcolemma over here. And we'll get to that. You'll see that in after a few slides. So more terminology. So axon terminal, we've looked at this in the previous picture. So this is the end of the axon. So the axon terminal and muscle fibers, they're separated by this gel-filled space that's called the synaptic cleft. So this area here, guys, the distance between these two, this is the synaptic cleft. And they're saying it's the gel-filled space. Now, stored within the axon terminals are membrane-bound synaptic vesicles. And these are these vesicles that I was telling you about, and they're filled with the neurotransmitter acetylcholine. Now, infolding of the sarcolemma, called the junctional folds, they contain millions of these receptors. So again, these are the junctional folds, and these are these ligand-gated channels. So you're going to have what they don't say over in that picture was. Hold on, where did I go? All right, yes, I think I went ahead. Anyways, let's go back over here. What they don't tell you over here is this: those junctional folds, they say they contain millions of acetylcholine receptors. These receptors, they're the ligand-gated channels. That's what they should say, ligand-gated channels. Now, the neuromuscular junction, this is important. The neuromuscular junction, it consists of an axon terminal, synaptic cleft, and a junctional folds. Right? It's a very good test question right over here. Does the neuromuscular junction consist of? Again, it's made up of axon terminal, synaptic cleft, and junctional folds. So let's look over here. Here we go. We've got your axon terminal, you have the junctional folds, and then you have this synaptic cleft over here. So again, this is filled with, it's a gel-filled space over here. So once again, what's going to happen is, let's take a look at this. You end up having an action potential that moves down. Okay. Now, as it's moving down the axon, it's going to terminate at this, the axon terminal. At the axon terminal, you have lots of these acetylcholine vesicles sitting over here, waiting to be released. When the action potential comes on, it pops open these calcium channels, and as the calcium comes in, it breaks these vesicles. So now you got acetylcholine that's moving around, and then it gets bound to the axon terminal, and then via exocytosis they get released into the cleft, and then they move across that cleft, and then they come to the other side where you have this junctional folds of the sarcolemma, and they bind to these ligand-gated channels. When they bind to the ligand-gated channels, what's going to happen now is you end up getting all the sodium ions that start to enter. Okay, and you get potassium that starts to exit, and this is what's going to cause depolarization. And it's going to generate this action potential now at the muscle, the muscle cell. So events at the neuromuscular junction, the nerve impulse arrives at the axon terminal, causing acetylcholine to be released into the synaptic cleft. acetylcholine diffuses across the cleft and binds with the receptors of the sarcolemma. And we saw this in the previous slide. We've already talked about all this already. Now, acetylcholine binding, it leads to electrical events that ultimately generate an action potential through the muscle fiber. Now, acetylcholine is quickly broken down by enzyme acetylcholine esterase. Remember, anytime you see ASE, you're looking at an enzyme. Now, once this acetylcholine esterase, once it breaks it down, now you get contraction to stop. Okay, so in other words, this happens so as acetylcholine comes in, right, it's sitting on top over here, sitting over here, sitting over here. Now you got sodium coming in. When the enzyme comes, it breaks this down, and then it causes them to get reabsorbed back up over here. So they'll go back up in there. But once that happens, then these guys close up, and now you have no more action potential, okay? So now you get contraction to stop. So once these guys are gone, no more sodium comes in, there's no more stimulation. There's not an action potential. There's no muscle stimulus. So the contraction will stop at this point, okay? That's all that they're trying to tell you over here. So this is the main job. This enzyme here is going to break down those the acetylcholine and ship it back to where it came from to stop contraction. Please be sure to look at the animation. If you go to the A&P the Pearson website for A&P flicks, again, there may be a DVD in your book, you can use that. And also, I'm sure there's a video of this on YouTube also if you search it. So again, this is showing you all the steps we talked about, and number 6 just shows you that acetylcholine esterase, it comes and it degrades the acetylcholine, and then these channels end up closing back up, so contraction stops. So again, this will get reused again. Acetylcholine, it gets recycled. They don't tell you that over here. Now, a lot of toxins, drugs, diseases, they interfere with the events at the neuromuscular junction. So, for example, myasthenia gravis, this is a disease that's characterized by drooping upper eyelids. You have difficulty swallowing and talking. And also, you just have this generalized muscle weakness. It involves shortages of acetylcholine receptors because the person's acetylcholine receptors are being attacked by their own antibodies. And this is suggestive of an autoimmune disease. I say that most of everything that we're going to be moving forward in this lecture, we've already discussed in the animations that we saw. So a lot of this is just going a little bit into more detail now. Resting sarcolemma is polarized, meaning there's a voltage exists across the membrane. So the inside of the cell is negative compared to the outside. Now the action potential is caused by the changes in the electrical charges. Now, this happens in 3 steps, and we're going to be looking at this in detail now. So we end up having this end plate potential, depolarization, and then repolarization. Now, for end plate potential, the acetylcholine, it's released from the motor neurons and binds to the acetylcholine receptors on the sarcolemma. Now, these are these ligand-gated channels that the acetylcholine binds onto, and this causes these channels to open up. When they open up, sodium, it diffuses into the muscle fibers, and some potassium, it diffuses outwards, but not too much. Now, because sodium is diffusing in, the interior of the sarcolemma, it becomes less electronegative. So in other words, we're saying it's becoming more positive. And this results in this local depolarization. That's what we call this end plate potential. When you look at this illustration over here, what it's showing you is this. Again, let's just recap. Remember, you have an action potential that's come down this axon. It comes to the axon terminal, and then that action potential pops open these voltage gated channels. As the calcium comes in, it causes these synaptic vessels to rupture and release the acetylcholine that is stored within. The acetylcholine will exit this axon terminal by exocytosis, then it'll move across the synaptic cleft and bind to these ligand-gated channels. Now, when it binds to these ligand-gated channels, it causes them to open up, and then you have sodium that starts to enter. Okay, and then this is when sodium enters, you get some potassium that starts to leave, and then you have this action potential that's generated. Okay, this initial action potential. Now let's go to depolarization. So this generation and propagation of an action potential is what's referred to as depolarization. And so if the end plate potential causes enough change in the membrane voltage to reach critical levels, called a threshold, then these voltage-gated sodium channels in the membrane open up. Okay. Now, once this happens, then you get a large influx of sodium that through these channels into the cell that triggers the action potential that's unstoppable. And this is what eventually leads to this muscle fiber contraction. Now, the action potential, it spreads across the sarcolemma from one voltage-gated channel to the next one and on in adjacent areas. And this causes the area to depolarize. So let's just look at illustratively what we just talked about. So remember, if we have here, so we're going to be going over, we looked at this step already, all right? Now we're going to be going onto this step over here, step number 2. So again, if there's enough of a change, all right, you have this, so if you have if the threshold is met, then this action potential, this wave is going to start to move across. And as it goes across, now right over here, we have these voltage-gated channels. All right. Now these voltage-gated channels, remember, if the threshold has been met, it's going to move across, and then it's going to pop these, open up these voltage-gated channels. And once this happens, then sodium starts to come in. So as the sodium starts to come in, what's going to happen? You're going to end up having the same thing that happened over here. Remember, over here you had depolarization that took place. However, it can only go so far. All right. So it just goes from here to there. That's as far as it can move. Now, once it moves over here, then, again, if it's strong enough, the threshold has been met, then it's going to move up to this point. When it moves over here, then you get more sodium comes in. And as more sodium comes in, guess what? You get further depolarization. Then as you move across, there's going to be another sodium-gated channel, a voltage-gated channel here for sodium. And that's going to, that action potential is going to move this way. And now this one's going to open. So more sodium comes in. And then you get another action potential that generates. So, and it just keeps moving on and on and on across. Okay, so this is the big picture that you need to understand. Initially, when the sodium ends up coming here, and if that threshold is met, that action potential is going to move up to here. And then once this happens, then more sodium comes and another action potential gets generated, or it helps to move forward. Now, repolarization, this is a restoration of the resting conditions. So sodium voltage-gated channels, they end up closing, and the voltage-gated potassium channels, they open up. So the potassium efflux out of the cell rapidly brings the cell back to its initial resting membrane voltage. Now this refractory period, this is for the muscle fiber when it's not able to be stimulated anymore for a set amount of time until repolarization is complete. Now the ionic conditions of this resting state, they're restored by the sodium and potassium pumps. Now sodium that came into the cell is pumped back out, and the potassium that's flowed outside gets pumped back into the cell. So if you look over here, again, we covered this step, we've covered this step. Now we're going to go into this third part, repolarization. You want everything to go back to the way they were. So what happens? Look, guys, remember over here, this is open. Now this is closed over here in this picture. So no more sodium can come in. However, now the potassium is, the potassium gate opens up. So now you can have potassium that's able to move about at this point. Okay, so now potassium can leave also. You can have an efflux of potassium that can exit. And now you end up having this repolarization that's taking place. And this time, this graph, it's just showing you the time it takes and the membrane potential. So again, you're going from minus 90 to depolarization that takes place. So if you look at it, by the time when the sodium channel opens up, which is probably, let's just say, right around, I would guess about perhaps 3.5-4 milliseconds to the time that polarization takes place, which is like less than perhaps between 1.5 to 2 milliseconds. And then repolarization takes place probably another couple of milliseconds later. This is how fast this occurs. You know, it's not even a second, but millionths of a second. Okay, that these things take place, from the channels open to depolarization to occur to repolarization starting again. So it happens extremely fast, extremely quickly. Now this excitation, contraction, this EC coupling. So these are the events that transmit the action potential along the sarcolemma. So excitations, they're coupled to sliding of these myofilaments, or this contraction. Now the action potential is propagated along the sarcolemma and down its transverse tubules, where you have these voltage-sensitive proteins in the tubules that open up to release calcium from the sarcoplasmic reticulum. And when the calcium is released, it is what's going to end up causing the contraction to take place. Okay, because it's going to move it's going to allow the actin sites to expose the binding sites for the myosin heads. So the action potential is brief and it ends before the contraction is seen. So here you go. So now again, what we're doing is we're kind of going back to look at this overall, to see the big picture, the real big picture of what's going on. So what was the whole thing? We want this contraction to take place, right? So what do we have? An action potential that moves down, and here, this is your the axon terminal here. This is your muscle fibers over here, and as the action potential comes in, remember you get these calcium channels here. They get opened up. Calcium enters. The acetylcholine vesicles break, acetylcholine exits, and then they open up these ligand-gated channels. Sodium enters, it creates this action potential. Action potential moves up along the entire sarcolemma, and if the threshold is met, of course, and then that action potential will travel down these transverse tubules. So this is where we're at. As this action potential travels down these transverse tubules, so first of all, remember, it's moving across the sarcolemma. So right over here, there's a T-tubule. As you're moving over here, you're going to have some more transverse tubules over here also, okay? And then you'll have more over here, and more over there, and more over there. So they're located throughout, okay, across this, the cell. Now, as the action potential is moving across the sarcolemma, it's going to come down this T-tubules, these transverse tubules, and it's going to open up these voltage-sensitive tubule proteins. Now, when these tubule proteins are opened up, it causes the calcium that's stored within the sarcoplasmic reticulum to exit. And as the calcium exit, it's going to come down and it's going to flood the sarcomeres. Now, when it comes to the sarcomeres, it's going to go and attach itself to the troponin. Now, when it attaches itself to the troponin then is going to slide itself off the tropomyosin. It's going to slide the tropomyosin off the binding sites of the actin. And now the myosin heads is able to bind to the actin to form this the cross bridge. So this picture is showing the same thing that I talked about before. Remember, as action potential moves along the sarcolemma and down the T-tubules, it's going to open up these voltage-sensitive tubule proteins, and it causes the calciums to exit. As the calcium exits, what's going to happen? It's going to go down and then flood the sarcomeres, where it's going to bind to the troponin on the actin filaments, and then the troponin is going to move the tropomyosin off the binding sites of the actin, so the myosin heads can go and attach itself there. Again, there is an animation. There is that shows you this process taking place. Please be sure to visit the AP Flix website or look at the DVD. And additionally, look on Google. You can Google it on you can Google it, or also you can go to YouTube and search this term over here, and you should be able to find it there. Now, this just kind of goes over everything we've talked about already. So it's just another review. And we're going to be seeing this over and over again. So when a nerve impulse travels down the axon of the motor neuron, when that impulse reaches that terminal, the voltage-gated calcium channels, they open up. Calcium enters the axon terminal, and then the calcium, it causes the influx of calcium. It causes the exocytosis of the synaptic vesicles that contain the acetylcholine. So when the acetylcholine is released, it moves across the synaptic cleft, and then it goes and it binds to the ligand-gated channels on the sarcolemma, and this causes the sodium channels to open up, sodium potassium channels to open, and this starts this end plate potential. Now, when the threshold is reached, the voltage-gated sodium channels, they open up, and that initiates this action potential, okay, to proceed throughout the sarcolemma. So again, they're showing you what we just talked about in the previous slide. You end up having an action potential that travels on the axon. Then when it reaches the axon terminal, it pops open these voltage-gated channels. As the calcium comes in, it causes these synaptic vesicles that contain the acetylcholine to release their contents outside of this and move across the synaptic cleft onto and attach themselves to these ligand-gated channels. When it attaches to these ligand-gated channels, now you have an influx of sodium that enters, and that causes this end plate potential to occur. Now, if this is strong enough, if you've met the threshold, then it's going to end up moving along this way. Okay, it's going to move along and then open up these sodium, the voltage-gated channels for sodium. And there we go. And this is what they're showing you in step number 2, step number 3, step number 4, and of course step number 5. These voltage-gated channels open up for sodium to come in, potassium to go out. And now we go over here to this part. So at low intracellular calcium concentrations, tropomyosin, it blocks the active sites on the actin. The myosin heads are not able to attach to the actin. So the muscle fibers, they remain relaxed. Now, voltage-sensitive proteins in the two tubules, they change shape. It causes the sodium, the sarcoplasmic reticulum to release the calcium into the cytosol. So at the higher intracellular calcium concentrations, calcium is going to go and bind to the troponin. This causes the troponin to change shape and moves the tropomyosin away from those the binding sites on the actin for the myosin heads to come attached to. Now the myosin heads, they'll bind onto the actin, and it forms this cross bridge. Now this is initiated, the cycling is initiated, causing the sarcomeres to shorten and muscles to contract. When nervous stimulation it stops, calcium it gets pumped back into the sarcoplasmic reticulum, and the contraction ends. So over here, again, we've looked at this already. So as that action potential is moving down, these voltage-sensitive tubule proteins open up, calcium floods out, and then as calcium moves, it goes to the sarcomere. It's going to end up binding to these to the troponin, and then when it binds to the troponin, it causes the tropomyosin to move off the myosin binding sites on this actin molecules. Okay. Now, once that happens, then the myosin head is able to attach itself to these binding sites on the actin. Okay, that's all that's being showed. So now, when they do that, we say you have a cross bridge that's formed. Okay, so contraction starts. Contraction begins at this point. Now, we're going to be looking at the 4 steps of this cross bridge cycle. So the very first step that we have is this cross bridge formation, and what we have here is this high energy myosin head. It attaches to the active side on the thin filament. The second step that we have is this power stroke, which is also known as this working stroke. So what happens here is the myosin head, it pivots and then it pulls the thin filament towards the M line. And the third step, the cross bridges, it detaches itself. And this happens when the ATP attaches to the myosin head. In the fourth step, we have the cocking of the myosin head that takes place. So what ends up happening here is that energy from the ATP hydrolysis, it ends up cocking the myosin head back into this high energy state. And this energy is going to be used for the power stroke in the next cross bridge cycle. So when you look over here, in this first step is showing you this cross bridge that's formed right over here. Okay, so what happened over here? Calcium came in, it attached to the actin, to the troponin on the actin molecule, on the actin molecule, which causes the tropomyosin to be moved off the binding site on this actin molecule, over here actually, and now this myosin head was able to attach itself there. Okay, so this is the first step, cross bridge formation. Now, in the second step, what ends up happening is you see that this the power stroke that takes place. So this ADP and the phosphorus, it's released. Okay, and when it's released, it causes the myosin head to pivot and bend, and this ends up pulling this actin filament towards the M line. So you see over here, it's getting pulled towards this M line. Okay, so it's going like this, and then as it cocks backwards, it pulls it closer to this M line. And now in the third step, you end up having this ATP ends up coming back and it attaches itself to this binding site on the myosin head. Now, once that happens, it ends up causing this cross bridge to detach. In the fourth step, what we have is this, you end up getting this ATP that gets hydrolyzed. So when it gets hydrolyzed, it energizes this myosin head, all right? And then this hydrolyzed myosin head, now it's ready to attach itself back out and get ready for the next power cycle over here, or the power stroke over here. So once again, please be sure to look at this animation on the Pearson website in the back of your DVD book, or you can go to YouTube and search it for it there. Now let's take a look at rigor mortis. So 3 to 4 hours after death, muscles they begin to stiffen. Now peak rigidity, it occurs about 12 hours post-mortem. So what happens is that the intercellular calcium levels, they increase because ATP is no longer being synthesized. So calcium, it can't be pumped back into the sarcoplasmic reticulum. And this results in these cross bridge formations. Now remember that ATP is needed for these cross bridges to detach also. Because you don't have that, these myosin heads, they stay bound to that actin. And you end up having this constant state of contraction. Now the muscles, they stay contracted until the muscle proteins, they break down, causing myosin to be released. So this is it for the first part for chapter 9. We have 2 more parts to this chapter to go. As I told you before, this is a long chapter. It's a very detailed chapter. Afterwards, it gets a little bit easier. On chapter 10, it's just a lot of memorization and just different muscles. And muscles are not that very hard to name, as you'll see when you get to that chapter. If you like this video, please give it a thumbs up. If you have any questions, please feel free to email me directly at my Gmail address. You can find it in the comments below, or you can send me a message through YouTube, or leave your question in the comment area underneath. Please be sure to subscribe. Please share with your friends, and best of luck. Thank you so much for watching.